Okay. Hi everybody, I'm Dave. Nice to meet you. I guess I could give a little bit of biographical information. I moved to the Netherlands at the start of last year. Before that I lived in South Africa for a while, but I spent two of those years working in Paris. But before that, of course, I'm an American. So I've been a little bit everywhere in the world, and I'm happy to talk with you about career paths in science, if that's interesting to you. But I'm primarily here to talk protein science and to talk about protein structure, particularly today. My background is not so much of somebody who's been hands-on instrumentation in the lab, but rather the person who figures out how to make sense of what the experiments produced in terms of data. So I'm a bioinformaticist rather than a protein chemist, but I've been spending all of my time in analytical chemistry labs. So you get a weird mix of the two from all that. We have a camera running up front. If you feel strongly that you do not want your voice recorded for something that's going to show up on YouTube, you can ask questions later or pass your question to the person next to you and have them bellow it out for you. Questions in flight are fine. They're actually very helpful to me to understand what you're getting and what you're not getting. And we have more seats up front, of course, so feel free to come on up if you like. There's nothing particularly holy about the front row. I am part of Ellen Nolan's laboratory, and I am, so now I'm working in aging research, obviously. And principally I'm now working with C. elegans worms, but over time I've worked in everything from soil bacteria to colon cancer, and tuberculosis for that matter. So I hope today to tell you a little bit of what I know about protein structure, because it's a discipline that's been moving so very rapidly that it seems like every year there's something very new to talk about. So I'm very excited about getting to talk about this. So let's start with a bit of dogma. I think you all know the central dogma that DNA makes copies of itself through replication. From DNA templates you can make an RNA transcript through transcription, and then you can translate that to produce proteins. Great. Central dogma, everyone should have that one. But what about this dogma? This is the dogma of structural biology. The sequence gives rise to a particular structure. That structure enables you to carry out a function. And this is very fervently what you would have received in the 1970s and the 1980s, and even a bit into the 1990s. But there are some things happening that are causing this dogma to be challenged. That would be the ideas about the intrinsic disordered proteins. And we're also going to talk a little bit about degrees of freedom in what kinds of structures are possible for a given sequence. From there, we're going to move on to talk about experimental determination of structures. This is not going to be really deep on the mathematics of diffraction or anything like that, but I thought it was worth noting that there are multiple paths you can follow to go from a protein to a known structure, a measured structure. And from there we're going to talk about the bioinformatics that have been changing so very much in the last few years. How do we predict protein structure given a protein sequence? So we'll talk about three different routes people have taken in that. Now, hopefully, this slide is not a whole bunch of new stuff. Hopefully, you've had some exposure to this in a protein biochemistry class along the way. So I like to start with something that I assume everybody knows, because most people feel comforted, frankly, to have a slide of stuff they already know at the very start. We're going to talk about plenty of new stuff along the way. The primary structure of a protein relates to the connection of the amino acids into a long polypeptide chain. This is what takes place at the ribosome. The primary structure is established there. The primary structure may ultimately be changed by the time you get to a finally functional protein. What if you've got a signal peptide on there or a propeptide? Stuff like that can get cut off, so that alters the primary structure of the polypeptide as it becomes a mature protein. Secondary structure represents these local interactions by hydrogen bonds that allow alpha helices and beta sheets to form. So the secondary structure is largely one of these local affairs that refer to whether this part forms a strand or forms a helix. That's not the only possibility. You might have a section of loop or a turn that appears in the sequence. So that's another kind of secondary structure. And then we get to tertiary structure, and I always find this one a little harder to define. Have people heard the term fold, a particular fold for proteins? Folds do not generally relate to a single helix or a single strand. They explain a relationship among multiple helices and strands. So in some respects, we think of this as kind of the fold, the structure that's adopted by the movement of all of these helices and folds and helices and strands to make the fold at that level. But there's more to a functional protein than just the structure formed by the polypeptide. You may have, in many, many cases, multiple polypeptide chains that come together to create the mature protein. So something like, say, alpha and beta hemoglobin coming together to create hemoglobin is an example of that. And it's also an example of the need for cofactors in some cases, because hemoglobin can't do its job if it's just a bunch of folded polypeptide chains. It also needs heme, and heme isn't coded for by the genome. This is something that gets incorporated into the protein to make it viable. And of course, the relationships, the binding interface between different polypeptides is quite important in how these quaternary structures form. So we have our cartoon of it over at the right, but this is, I hope, a useful reminder of all these things that result in protein structures. Now, I kind of like to remind people that science is not something that started in your lifetime, and nor is it something that started in mine. This is a letter to science written by a Nobel Prize winner in 1973, which coincidentally is the same year I was born. So let's talk about the thermodynamic hypothesis. This is kind of an old school way to think about structure. The thermodynamic hypothesis is that the 3D structure of a native protein, native here meaning folded up, in its normal physiological milieu. Anyone want to take a guess at what normal physiological milieu means?
Environment.
Like sort of their normal environment?
The environment.
Yeah.
Okay, so what factors come into play to create the environment of a protein? pH, very good one. Sure, is it acidic, basic, neutral?
Temperature?
Temperature, yeah. Definitely proteins take on very different structures with temperature.
Humidity.
Sorry?
Humidity.
The humidity. Well, I mean, it's an aqueous environment, hopefully. But there's something connected with the aqueous environment that I think is really worth talking about. The movement of ions through boundaries matters, but more specifically, hydrophobicity. You may have an environment that's just aqueous, like an in-solution protein, or you might have something that lives in a membrane. So these all take a part in creating the normal physiological milieu. Okay, so the structure that they adopt is one in which the Gibbs free energy of the whole system is lowest. I always found Gibbs free energy a little bit puzzling as a biochemical concept, but I think of it in this case as at rest. What does this protein look like? You know, you might kick up you might kick up the temperature, you might alter its pH away from the normal physiological milieu, and suddenly you have a very different structure at play. So at rest, this protein takes on this conformation, and then the native conformation is then determined by the totality of interatomic interactions, and hence by the amino acid sequence in a given environment. So the totality of interatomic interactions is not limited to the polypeptide itself. The very obvious thing that you have to take into account is the presence of water. Water is certainly going to play a lot of roles in stabilizing different parts of the protein structure. So the interatomic interactions then need to involve the relationship between the amino acids themselves and with the milieu. Okay, I've said milieu enough times for this year, so I think we can just cross that one off the list and call it finished. All right. But there are some big things that we need to be careful about. It's easy, I think, when you flip through a biochemistry textbook to see this is the structure of hemoglobin, this is the structure of a transmembrane domain, this is the structure of all. Those are snapshots, right? They're snapshots of a static, fixed shape for a protein. That's not how proteins live. So let's read this quote. Now we're up to 1983, still long before your time. Do you remember 1983? Yes, 1983 was a pretty cool year. All right, so protein structures are not static. For times up to a few tenths of a picosecond, the motion of individual groups in a compactly folded protein is therefore similar to that of small molecules in a liquid. The groups jiggle about. Did you know you can use jiggle in a scientific paper? You totally can. The groups jiggle about as a result of frequent collisions with the surrounding cage atoms. Boy, that's a really epic term for just a bunch of water that's sitting around. But yeah, okay. So over longer times, the displacements are limited by the forces that maintain the native folding pattern of the protein. So this jiggle about is just going to happen by the thermodynamics of the motion at the molecular level. But there's some constraint on that. You're not likely to rip apart a hydrophobic bonding interface just because the molecules of water around you are a little warmer. A degree C is not going to make that big a difference. The net motion of a typical group is therefore a superposition, it's an additive effect, of the rapid local jiggling and the slower elastic movements that involve larger regions in the proteins. So we have a dynamism to these proteins. We have some amount of jiggle that may or may not play a physiological role in how it functions, but we also have larger motions that reflect the activity of this protein. Okay, so this is 1983. And finally, we get to this even bigger challenge, and this is one that I still haven't got my head wrapped around, even though it's been in the works ever since I was in college, right? So, well, 1999, I was in grad school by that time, to be fair. All right, is the native structure always enough to understand the function of a protein? Although the functions of many proteins are directly related to their three-dimensional structures, many proteins, numerous proteins that lack intrinsic globular structure under physiological conditions have now been recognized. Such proteins are frequently involved in some of the most important regulatory functions in the cell, and the lack of intrinsic structure in many cases is relieved when the protein binds to its target molecule. If I have a dog, what is the structure of the leash? Normally it's just hanging on a peg in my coat room, right? But the minute I have the dog there with its collar, and I've clipped the leash to its neck, and we start walking around the sidewalk, suddenly it does have a structure, right? The ligand is there, and now the structure is able to take its form. If you have a protein that requires ATP to undergo its activity, then it may be that the part that binds the ATP is not particularly structured until ATP is in the mix. So you may have structures to these proteins that only emerge when its binding partner is there. Okay, and I believe that Elizabeth would definitely want me to say that sometimes there is no formal structure that ever really results from a part of a protein, and yet it may be functional. So there are ways to continue this discussion on to all that we know about intrinsic disorder in the last 25 years. There are a lot of things that have changed. I said I wanted to talk about degrees of freedom. Does everyone remember a biostatistics class? Okay, so we're going to now arch over that concept of degrees of freedom from statistics to degrees of freedom for protein structure. What's changeable about a particular polypeptide that allows it to take on different conformations? For that, we need to talk about phi and psi angles. We have a phi and a psi. Everyone can tell me where the peptide bond is in the structure, yes? Actually, we have two of them. Everyone sees them? What is the characteristic of a peptide bond? It's connecting amino acids, yeah? What's the backbone sequence, or the backbone letters that you find for the elements that form a backbone of a polypeptide? Do we have any Star Trek fans in the room? Any Star Trek fans? No one likes Star Trek? Oh, this is harsh. You're killing me, guys. All right, well, I like to remind everyone that the call sign of the Enterprise is NCC 1701. And if you ever forget what atoms make up the backbone of a polypeptide, you just remember NCC. So here we have NCC, NCC, NCC. We have 3 amino acids here, therefore we have 2 peptide bonds. Does anyone want to admit to watching an episode of Star Trek at this point? Oh, okay, that's fine, that's fine. All right, so we have a peptide bond here and a peptide bond here, but neither of those has a little turny arrow associated with it or a Greek symbol stuck on it. Why is that? Because in most cases, a peptide bond is going to have the oxygen of the carbonyl pointing one way and the hydrogen of the nitrogen pointing in the opposite. This is kind of a fixed plane for that bond, basically. So there's not a lot of rotation there. But we do see rotation between the nitrogen and the alpha carbon right here. So the spinning of that bond is referred to as a phi angle. Between the alpha carbon and the carbonyl, we have another turning point. This is the psi angle. Okay, so you could imagine then that if we have some free rotation here and some free rotation here, we have 2 possible turning points per amino acid in a polypeptide. So if I have 100 amino acids in a polypeptide, how many degrees of freedom, how many angles can be turned within that 100 amino acid stretch? Each amino acid has how many spinning angles? 2. So 100 amino acids in a chain have how many turning bonds? 200. 200. Okay. So we can start to talk about why this is daunting in just a moment. But for now, just remember that phi and psi angles represent the nitrogen and the alpha carbon and the carbonyl spinning. So, is it possible that every amino acid allows for spinning around this particular bond? Can anyone think of a particular amino acid side chain that would block the free spinning of that bond? Yes?
I'm not sure, but maybe if it's very, very bulky, then it's less likely to spin.
Okay, so something like tryptophan or tyrosine, phenylalanine. That's one possibility, but not the one I'm thinking of at the moment. Yes?
Protein?
Proline. Okay, why is proline suspicious in this case?
Because it doesn't have a side chain. Or it doesn't have what's it called again? The residue? Or, I don't know how to say this.
Okay, well, so he's raised proline as a suspicious character for the phi angle. For the phi angle, why is proline unable to spin its phi bond? Yes?
Is it that it binds back on that nitrogen?
Correct, yes. The side chain of proline, this R group here, actually swings all the way around and replaces the hydrogen on that nitrogen. So, because there's a ring there, it obviously can't spin. Okay? So, this is one of the reasons why proline is always going to be a special character in the world of protein structure. It's not even really an amino acid, it's an amino acid. If you remember your biochemistry, it's a funky thing. So proline has a lot of weird characteristics that make it kind of a standout in protein structures. All right, and that brings us to Ramachandran plots. If you're trying to understand protein structure in the main, you probably want to understand which amino acids, which phi and psi angles are commonplace and which are rare. So let's start with a phi angle of 0. Here's phi down here, it's very, very small. 0 is this line drawn right down the middle. And we see that there's no density there. There's essentially no incidence of amino acids spinning to a phi angle of 0. Why is that? The reason why is this steric distortion. Does everyone remember the word steric from biochemistry class? All right, so, or if you took physics, the physics route here, then maybe you remember the Pauli exclusion principle, that you can't have two atoms that are trying to exist in the same space. So if you turn a phi angle to 0, you end up having the 2 carbonyls fighting each other. And this is not something that electrochemically is going to be very viable. So a phi angle of 0 is almost never observed. But what we see is that what kinds of phi and psi angles you have is a pretty good hint about the secondary structure that we observe at that point in the protein. So we have right-handed alpha helices, left-handed alpha helices, and anti-parallel beta sheets that have these characteristic phi and psi angles. So if you know the structure of a thing, you can compute what the phi and psi angles are at any point in that structure, and you can then ask, do they fall in this region occupied by a particular, that is commonplace for the bond angles of a particular bit of secondary structure. It's a hint, at least, at what the structure is there. So at base, we know that there's a lot of room for possible conformations for any one given polypeptide sequence. You could imagine lots of different structures, some of which have high probability and some of which have nearly no probability. I mean, certainly any structure that brings two chains to try to occupy the same physical space, that's not allowable, right? Any more than with your shoestrings. So Leventhal put together this hypothesis that I thought was, this paradox that I thought was a useful expression of that. If you have, in the case of our 100 amino acids, 200 bonds that can spin, and each of those could adopt, let's say, let's treat this kind of like the hands of a clock. Let's say that each of the phi and psi angles is allowed just 12 different possible positions. How many possible conformations are possible for a 100 amino acid polypeptide, if every possible bond angle is feasible? Well, we've said we're going to have 12 different possible positions for each angle. We've said that we have 200 possible turnable angles, turnable bonds within the polypeptide. So 12 raised to the 200 is a very, very large number, right? This is what Leventhal was really worried about. If the search space is really close to infinite for finding the right structure, how is it that a protein, upon being translated, a polypeptide, upon being translated, knows, or if these proteins are just sampling the space, how is it possible that they come to the right fold in the end? It's a big question. So it wouldn't be possible in a physically meaningful time, if they're just sampling randomly, to do that. Do we require angels to come down and bless each polypeptide as they come off of the ribosome? This is not how it works, actually. So instead, what we see is that there's a small energetic bias. Each of these bond angles is not just randomly rotating around until they click into space. There's a small energetic bias that says, well, this set of bond angles is going to be a lot more comfortable for this part of for this amino acid than the others. And so at rest, it will come to the correct angle in most cases. Are all proteins in our cells in the correct conformation at all times? No. No, they're not. In fact, some really profound diseases can result from proteins that take on the wrong fold and encourage other proteins of the same sequence to take on that same wrong fold. Stuff like mad cow disease results from that. Those are prions and things like that. So our physiology and that of all the other living things out there rely on these very small energetic biases that allow little bits of the sequence to fold up correctly, and once they're correct, there's not a lot of impetus for them to try sampling new conformations. And by these small pieces folding up correctly, the larger pieces can fold up correctly as well. So how do we go about measuring a protein structure? I'm realizing it's going to be a little distracting that I don't have a clock to look at in this classroom. I can talk for a long time about a single slide, so I have to be careful about that. Perfect, thank you. So let's start with the big one. X-ray crystallography is our best friend in the world of protein structures. At the moment, I think something like 80% of all the structures in our databases come from X-ray crystallography. So let's walk through it. What is the crystal in X-ray crystallography for protein structure? Yes?
I think you take a bunch of the same proteins.
Yes?
And then they form sort of a shape which is very structurally organized.
Yes.
And that sort of is the crystal because they're in a certain order.
Full points. That's really good. So yes, we have a bunch of copies of the same protein. Those proteins crystallize with themselves, and that forms the crystal that we're going to use. Do all proteins crystallize equally well? Definitely not. Membrane proteins have been bedeviling the X-ray crystallography world for a long time, and they've found some nice workarounds on it, but generally speaking, we're talking about a protein crystal that's more or less homogeneous. It is plausible, though, that the protein structure you really want is not pure for a single polypeptide. Could someone come up with some reasons why? Under what circumstances would you want something else in the mix? Let's take an example. Oh, yes?
Well, if you have, for example, protein that you want to crystallize, but it cannot crystallize by itself, then you can use another protein which can crystallize, and then you'll have two.
Right, right. So it could be that the protein's native structure is really only achieved in combination with other polypeptides. So if you were trying to create a crystal of hemoglobin, trying to do it with just alpha hemoglobin alone would be kind of silly. You'd expect to have both, because its mature conformation requires both chains. It's also the case that very frequently one part of the protein is very differently crystallizable than the other. If you have, say, a protein that's normally moored in a cell membrane, you might want to lop off that piece and get just the part that's in solution and try to get that to crystallize independently of the rest. It might be that you need some non-protein pieces. You might need heme, for example, to get things to fold up properly with certain oxygen ferrying proteins. So there are lots of different things that could cause what we subject to X-ray crystallography to be both more than and less than what the sequence of an individual polypeptide is all about. Okay? So having formed these crystals, which is frequently a process that involves several graduate student careers, you finally can get to a crystal that you can send to a specialist facility that can hit it with X-rays. Are they just taking it to the local dentist's office? No, there are relatively few sites that actually specialize in crystallography of this type. So we're not doing an experiment that you can just do in every lab. You might be taking it to a local facility that specializes in that sort of thing. All right. We also have this problem of diffraction. So we've said our crystal, this big blob up here at the top, is formed from the protein plus or minus some stuff. We've got that crystal. We're sending X-rays at the crystal. And now we see that their paths are bending. This is the process of diffraction, that the crystal redirects a beam of X-rays to a new angle. And over here, we have the detection of those X-rays after they've diffracted through the protein crystal. So we will talk a little bit on the next slide about how we interpret these data. It's actually pretty messy and confusing if you ask me, but I hope that we can come to at least a useful understanding of how these data enable us to get structures. It may be that your protein is nice and small, and it might be that your protein is dynamic enough that you need to get sort of a picture of multiple snapshots, if you will, of how those proteins are behaving in solution. And for something like that, NMR is a pretty good option. We see that the resolution at which we know these structures gets a bit worse as we move from X-ray crystallography to NMR, but it's still good enough in some cases that you can put together a useful structure, even multiple snapshots of structures through NMR. And it might be that you have some massive complex to deal with. And for that, you might need to use something like electron microscopy. So when you're looking at protein structures, it's always a good idea to keep in mind what technology gave rise to it. Did this come from X-rays? Did this come from NMR? Did it come from electron microscopy? Could it be that we are altering the structure of proteins by forcing them to crystallize. As such, it might be that the X-ray crystal structure that we have for a protein is not, in fact, has some important differences from the active form of the protein. So keep in mind that any static snapshot of a protein structure is missing out on the dynamics of how that thing behaves in the physiological environment. Okay, gonna move ahead. That's interesting. There we go. So, algorithms infer structures from electron density. Remember, we started with a crystal, we then produced diffraction data from it, and now we have an electron density map. There's something missing when you look at an electron density map for a particular protein, and that is phase. The phase refers to more or less where within the structure the collision takes place, and so you can end up with a mix of these phases of the data for the electron density map. So you have to reconstruct that phase information using some of the information we've learned from long experience with X-ray crystallography. But at the end of the day, we end up with a density map to say that within this 3D structure for the unit cell of the crystals, we have this notion that something here is causing electrons to diffract, something here is causing the X-rays to diffract, and so on. Those don't come with labels, and depending on the resolution of the thing, you might find that you have some places that you're very confident where there's something causing X-rays to diffract and things that aren't. The shoulder on that might be a little confusing. So we have some parts of the structure that have very good clarity and some parts that don't. So backbone tracing is one of the first things we're going to want to accomplish, and hopefully there's a lot of software to do this for us. We have some putative sequence that this crystal represents, and that sequence should be traceable as an NCC, 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 NCC throughout this 3D space. So figuring out where the backbone of the polypeptide is kind of the first bit of business to do. And only after you have this trace of where the backbone runs throughout this 3D space do you start to place where the side chains are and how they're oriented relative to that backbone. And I read this joke on Twitter a few years back, and I haven't really stopped loving it ever since. So what did Watson and Crick do all the X-ray crystallography on DNA? They did not. But they did get early access pre-publication to what Rosalind Franklin had done on the DNA electron density. So they got Nobel Prizes and she didn't, right? Not very cool. This stuff happens too often. All right, so I'm not going to drive you through a whole bunch of math on how X-ray diffraction works in this context. Frankly, because I don't understand it that well myself. We all specialize in something in science, and I did not specialize in interpreting electron density from diffraction. So we'll step onto the protein data bank instead. Protein structure is kind of a new thing, right? No, we've actually been doing protein structures for a very, very long time, and the protein data bank has been collecting this type of information since 1971. So each of these is going to get a 4-letter accession within the protein data bank. It's not huge when we look at how big the protein data bank is by year. We're up to what, 250,000 structures? How many different polypeptide sequences do we have?
I think we have around 20,000 genes, right? So.
Oh, for humans?
Yeah.
Okay, yes. So I wasn't limiting to humans here, but
Okay.
okay, so yes, a human has 20,000 genes. Do we have 20,000 proteins?
Almost, I think, maybe.
Does anyone want to agree with that or disagree with that? Do we have 20,000 proteins?
No, because of splicing.
Splicing, very important thing, yes. That's definitely a great thing to think about. So because we have different ways to stitch together our exons, a single protein coding gene can lead to multiple proteins. Is that an everywhere thing, or is that relatively few of the proteins we have? Okay, I download the human proteome for identification of proteins very, very frequently. And I can tell you that today, if I download a sequence database from Uniprot representing each protein coding gene, reporting each well-documented exon shuffle, I get about 100,000 sequences. So on average, a given protein coding gene leads to 5 different spliced sequences. Okay, so if we were just counting human proteins that represent different amino acid sequences, I would say the current count is around 100K, even though we have 20,000 genes. But in this case, we're not talking about just human proteins. We're talking about proteins for soil bacteria and yeast. And I mean, yeast, everyone loves yeast, right? Yeast was such a great system. We got so much good stuff out of that. But over time, these 250,000 protein sequences, 250,000 protein structures have come to represent a huge diversity of folds from a huge number of bacteria and a huge number of archaea and a huge number of fungi. And we have a huge diversity of structures now available to us. Do we have a structure for every human protein? That's a big no. We definitely don't. We're not close to that either. So what we have are the structures that we can get from the species that we do care about, and yet there are some things that have proven quite resilient against our getting a decent structure from them. All right, so each of these is going to be a structure that's associated with X, Y, and Z coordinates for each of the atoms within the structure. In some cases, there will be some points of vagary within the structure, and some parts that are absolutely, certainly at the right position, and some that may be a little more flexible. What we see is that the X-ray structures end up comprising 81% of the total at this point. It's a very, very dominant technique for this. NMR and electron microscopy have also contributed a lot, but electron microscopy has been catching up a lot. It used to be that NMR was our second biggest contributor of structures, but now it's EM. So that's been a big difference in the recent years. And a resolution below 2 angstroms is common. So our ability to place an atom within this 3D structure is very precise, I would say. It's remarkable that that's achievable. One of the things I would have you take away, though, is that the number of sequences we have across all of biology is vast by comparison to the number of structures. 250,000 is not even a scratch on the surface of the set of protein sequences we have today. The number of sequences we have from the number of species we've investigated, I mean, I assume we're in the billions at this point. It's just incredible how quickly a novel organism can be sequenced to produce lists of putative protein sequences. We don't have structures for even a very small number of those. So, what do you get if you go to the protein data bank and download a PDB file? A PDB file is all the information relating to one of these 4-digit accession codes. So here I've grabbed a structure for 1BEN, and I've grabbed out just a very, very small part of the PDB file that results. These are atom lines. As you can see, they're all marked atom at the start of the line. And we have what atom do they represent within that structure. We have what part of an amino acid do they represent. What is the 3-letter code for the amino acid at that position? Which polypeptide does this one belong to? Where does that amino acid fall within that polypeptide sequence? And then we have this information over here. If I remember correctly, these 3 columns are the X, Y, and Z coordinates for these individual atoms within the structure. Normally, when you look at PDB, you're not looking at it this way, are you? You look at it and you get this beautiful structure out, and you maybe poke it with the mouse and it rotates around for you and it does all the nice stuff. But it's doing that based on this information. So I would like to remind you that when you download a PDB, you're not downloading a picture, you're downloading data. And it's in a text format. You can just open it in Word if you really felt like it. I mean, that'd be awful. Open it in a text editor instead, but that's fine. These are the kinds of data that fall out of that. And there's a whole lot of annotation information explaining what publication is associated with each of these structures. There's a huge amount of information buried within these. we show a lot of structures in our journals, and I think it's worth taking a moment to talk about different visualizations that could be made from them. It used to be that you would need to have a fancy Silicon graphics machine to do all your visualization of these structures, but we've long since moved past that. Your web browser, your phone can very happily visualize these structures for you today. But I think it's useful to talk about these 4 different structure types that are available when you go to RCSB and produce this visualization. We start with the cartoon. The cartoon is a really common way to visualize structures because one of the first things you're going to want to know when you look at a structure is which parts of this are a helix, which parts of this are a sheet. And that information is made available this way. So you see these long ribbons that have a little arrowhead on them. Those are beta sheets, or strands that are going to be part of a sheet. And the helices have this nice curly ribbon, like if you were wrapping at Christmas time and you hold the ribbon between your thumb and an index finger and tightly rip it, you'll see it do the nice corkscrew thing. That's our helix symbol. Okay, so we also have some parts that are indeterminate, right? The bits of polypeptide that are connecting a helix to the next helix, or a helix to the strand, those are just shown as kind of these curving lines without much of a cartoon shape to them. So you're already getting this kind of 3-state appraisal of this polypeptide. Which parts form helices, which part form strands, which parts form turns between them. So the cartoon gives us that information very compactly. Do you see a number of sprinkles there? A few sprinkles on that figure. Those are parts of the protein that, these are parts of the structure that may represent things like water molecules, for example. So not everything is part of the polypeptide, therefore it's not connected to a backbone. The molecular surface depiction of this structure is an attempt to something you might see called space fill sometimes, a space filling visualization. It's an attempt to show how the bulk of not just the backbones but the side chains form the outer surface of the protein. So for that, a molecular surface diagram is more appropriate. Maybe all you really care about is what kind of twists and turns the backbone makes within the structure. And for that, you can get a lot of clarity by just using a backbone visualization of it. Now, superficially, the ball and stick diagram might look very much like the backbone, but there's a very important addition here, and that is, this is showing side chains as well. So all that hair that you see growing off of the backbone, those represent the amino acid side chains that are giving this thing greater body than it would have otherwise. If you look in very closely, you can see why it's called ball and stick. It's because the nuclei are represented by balls and the bonds are represented by sticks. So looking at this more closely, you would see the namesake for it. It's very commonly the case that if you're producing a figure of this sort for a poster or for a paper, that you're particularly wanting to draw attention to these 3 amino acids, for example, or show how the structure would vary if you replaced one amino acid with another. So for that, you're likely to maybe have something like a backbone visualization for which you've turned on the ball and stick visualization just for the side chains that matter. That sort of thing can be very helpful. If you're trying to show how a ligand interacts with this protein, you're probably more likely to use a molecular surface depiction of it. But you will sometimes see that different amino acids are visualized using different strategies to help your visualization in a particular case. And I chose the case of gluten because someone dear to me has celiac disease, and gluten is a real pain in our backside. So it's a worthwhile protein to think about the structure of because it can cause a lot of grief to people. Okay. Now, I just said we had 250,000 structures. Are those 250,000 structures all completely dissimilar? No, definitely not. There are common folds that we see, and it may be that a protein you got, say for pyruvate kinase, might have a strong evolutionary history with a pyruvate kinase in a blade of grass, or in wheat, or something like that. So you might see that although your sequence for pyruvate kinase and the blade of grass's sequence for pyruvate kinase have had a lot of time to diverge from each other, they still adopt a similar fold. So people who've invested a lot of time in this, the folks at SCOP and the folks at CAF, have really focused on trying to detect either by human curation or by automated curation which of these structures fall into the same fold. Now, I said before that fold is a term that gets used a lot in this field but can mean different things to different people. So in this case, we're going to start with CAF over here on the right. So CAF was, its big publication was back in 1997. They have class, architecture, topology, and homology. If you take the first letter of each of those, what do you get? CAF. So in the name of the software, they're telling you what are the layers that they're using for expressing the commonality of different folds. So we start with mainly alpha, mainly beta, alpha, beta, and a few SS. I don't even remember what that one stands for. A few strands, perhaps? I don't know.
Secondary structures, maybe?
So, what's that?
Maybe secondary structures?
Yeah, that could be. It could be that you're dealing with sequences with relatively few secondary structures, and having a place to categorize them would make sense. Yeah. So in this case, just because two structures fall in the same class doesn't mean that they're really, really similar, but it means that they have at least the same kinds of secondary structures contributing to their overall structures. Within that, within each of these four, we have an architecture. Within each architecture, we have a topology. Within each topology, we have homology. So you might have multiple sequences that all fall in the same basic fold, and then those would all represent the same topology, same architecture, same class, just differing at the level of homology. So CAF makes it possible to look at these structures and say, which other structures in this database are most similar to this one? Maybe you'll find other sequences that have adopted the same fold. And maybe that's because of divergent evolution, but it might be convergent evolution as well. It is certainly possible that sequences can drift to become very similar to other folds as they over the course of evolution. Okay, so that's CAF. Now over here at the left, we have SCOP, and this is not the first publication for SCOP in 2014. They were published much earlier than that. But I thought this was important because they represent kind of changing paradigms in how biological information gets classified. So we start with its origin as it manually curated categories of structure groups. And that reflects basically how PDB started as well, that an expert was looking at the structural data and deciding it deserved inclusion in PDB. And in the same way in SCOP, they said, well, we have things like beta barrels. Which structures fall within this category of beta barrels, or leucine zippers, or whatever structure you want to name? So SCOP begins with manual curators. And it begins also with things that fall into this class. Okay, so I have a category of things called beta barrels. What structures fall into that class? But over time, they decided that was not actually the right way to proceed. They shifted away from this hierarchy of different classes and instead began looking at direct structure relationships. So they're moving away from a these are beta barrels approach to these structures are the two most similar to each other within the database. Therefore, they bear a direct relationship to each other. So what you see coming out of this is no longer that you go looking for the beta barrel box. You instead say, I have this structure in which I'm interested. What other structures are most similar to it? What you have is a graph that comes out of this. Do you see what I'm talking about? That for each structure, I can now say what other structure is most similar to that. I can ask, for example, for two structures, how many intervening structures must I move through to get from one to the other? You can find shortest path algorithms on it. So we move away from a how many structures fit in this box paradigm to which are most closely related, how many structures do I need to walk through to get from one to another. It's a graph representation rather than a box approach. And then more recently, SCOP has realized that the primary users of SCOP are not generally going to the SCOP database and poking in a uniprot accession to get a structure back. Instead, what they realize is that SCOP is now used as an input to all kinds of other databases. So if you go to Uniprot knowledge base and you go searching for a particular Uniprot identifier, you will also find, in all likelihood, if a structure is known for it, a SCOP classification. And it makes it very easy for you to become a user of the SCOP database without ever once going to the SCOP website. So SCOP shows up everywhere because they've made this really strong emphasis on making their data accessible to others. Okay, so given structures, we can infer something about the commonality of different folds. CAF has the class architecture topology homology structure for these things. Here we see the family, the superfamily, folds, and classes. Different names, but for kind of the similar concepts. But at this point, we're going to shift away from making sense of experimentally determined structures, and instead ask, how can we go about inferring a structure if what we have is a sequence? Okay, what's our time check? Perfect, okay, good. So, I'm going to start once again with something old, and then we'll talk about something new. So, Chow and Fassman published they were working on this stuff as I was born. They published it the year after I was born. Chow and Fassman were concerned with how can we know which parts of an amino acid sequence are likely to form helices and which parts are likely to form strands that are going to be involved in sheets. So for that, they decided they would create sort of a helixness weight for each amino acid and a sheet, a strand weight for each amino acid. So some amino acids are simply more likely to be found in alpha helices, others are found more likely in sheets, some are not likely to be found in others. So something like proline, for example, doesn't have a huge degree of helixness or strandedness because it shows up in turns all the time. So I would also say that they have, they split the set of amino acids into alpha helix initiators as the things that are most prone to being in helices, and helix enders as the amino acids that are most likely to end the helix part of a protein sequence. So for this, they have this concept of a nucleus for the structure. So we're not talking chemistry now. We're saying that within the sequence, we have a region that's really dense with amino acids that are very friendly to helices. I believe they said that they needed 4 out of 6 consecutive amino acids to be on the helix-friendly list to create a nucleus. I think they said you need to have 3 out of 5 consecutive amino acids to have a nucleus for a strand. And then from that nucleus, you can expand out in either direction until you hit helix-terminating or strand-terminating amino acids. So you look for the spot that's really rich in alpha helix friendly parts. You extend out this way, suddenly you hit a proline, you're at the end of the helix in that direction, you come back this way, and you hit a proline in this direction, so this is now your helix bound. I'm highlighting proline too much, it's just an example of a helix breaking amino acid. Okay, so those nuclei can then be expanded out to say, this is where a helix is found in the sequence. How did they come up with those numbers, do you think? Did they toss it into machine learning and see what happened? No, because they didn't really have that available to them at the time, not in the 1970s. So instead, they used what they knew from biochemistry, from the existing set of structures, which was more like dozens at that point, not thousands, and they used what information they had available to them to empirically come up with scores that seemed about right. And so Chow and Fassman became the standard way to predict secondary structure for a long time because of this. But more recently, we've seen tools like JPRED have benefited tremendously from the onset of machine learning. Machine learning is what preceded AI and what will remain after AI is dead. Sorry, somebody has to say. Machine learning is vitally important across all of bioinformatics. It's very difficult to find any part of bioinformatics that has had no touch of machine learning over time. And this well precedes the advent of LLM and all, large language models and stuff like that. So machine learning is hugely important in all kinds of signal detectors built into bioinformatics. So JNet 2.3.1 was an effort based on these SCOP classifications to find which amino acids in these folds have which secondary structures, strands and helices, within their sequences. Having trained the neural network on that selection of scope sequences, it was then able to say, for any given position within the amino acid sequence, is this an alpha helix, is this a beta strand, or is it a coil? I use the term coil rather than turn, but it's same concept really. So it might be that you have a particular sequence that you're interested in, there's no decent structure for it, and so you're just feeding it this one sequence. It might be, however, that you've studied this sequence across several different species falling within the same genus, in which case you might have this nice multiple sequence alignment to show what variation occurs across this polypeptide sequence. So you're not limited to saying one sequence at a time. You can feed it a multiple sequence alignment as a way to capture the variation in that structure. And that will give you a more nuanced appraisal of what structure comes out. Naturally, predicting secondary structure alone is not the only thing that we want to do. It's really very valuable to us if we're able to start from a sequence or a multiple sequence alignment and just predict the full structure. Not just to say this is a helix and this is a strand, but this is the fold that they adopt when this entire sequence folds up. Now that's a much harder proposition. So I would start us back in 1997. In 1997, there was a remarkable group at the University of Washington doing protein structure analyses unlike anything I'd ever seen before. David Baker was the guy who taught me protein structure back at the University of Washington, and he was also one of the leading lights of this toolkit called Rosetta that became the standard for how to do de novo sequence inference, de novo structure inference over time. So it has a lot of different concepts that show up in everybody's work, basically. Everyone was using some of these elements in the other tools. Now, I'm going to talk about AlphaFold next, which has been like the biggest story in structure for a long time. But it's important to remember that there are decades before that in which protein structure was being predicted as well by these methods. So let's start with rotamers. It's very commonly the case that when you read about the algorithms people are using to predict structure from nothing, from just the sequence, that they talk about the use of rotamer libraries. And most typically, this means we have a very large collection of 9 amino acid segments from known structures, and we know what fold these adopt typically when we see them in structures. So something like www, www, www is not seen very commonly. Tryptophan is not a common amino acid. But there are plenty of amino acid motifs that we see that are about 9 amino acids long that have occurred in lots of structures, and we have a pretty good sense of how their backbones look if you average across many, many structures. So this is called a rotamer library. Anytime you see this set of 9 amino acids, you have a guide to what kind of structure they adopt because you've seen them before. You've added that information, encoded that information into a rotamer library. Okay, so use what you've seen before is a very good strategy, I think, for anyone trying to come up with something new given a random sequence. Have people seen the term Monte Carlo used in connection with bioinformatics? Huh. What is the thing that you go to Monte Carlo to do? To gamble. To gamble. It's a great place to go gambling, good casinos and stuff. If you've watched enough James Bond movies, I'm sure Monte Carlo has shown up plenty of times for you. And in the same way, Monte Carlo is a bit of a game of luck in bioinformatics. That we have some set of phi and psi angles available to us in this protein sequence, and we're going to guess that they have these random positions, nothing that actually leads to collisions in space, but we can give it some set of phi and psi angles that is at least plausible, and we can use that as a starting point. So let's imagine that we have a whole fleet of computers available to us. We have a cluster. Maybe we have 200 nodes, each one with 12 cores available to us. So we have a lot of cores that we can throw at this problem. We might try 10,000 possible starting points for this particular protein structure and give each of them to a different processor on a different node and ask it, how well does that refine out? What's the Gibbs free energy associated with that particular structure? Now, are any of those 10,000 going to be exactly right? No. But having thrown out 10,000 possible models for the sequence, some of them have relatively low Gibbs free energy, and some of them have relatively high Gibbs free energy. You can then say, throw out everything but the top 10% of my starting positions, and now make a new set of 10,000 starting from the winners of the last round. This is called genetic programming, by the way. So you can start by creating a particular posture for what this sequence is supposed to look like, one of these 10,000 starting positions. You can specify what you're going to alter about that sequence, about that structure that you put forward. You have a scoring function that evaluates whether this is a plausible structure or not. And from that, you can choose which new poses you're going to start from to see the next generation of models. So we have an iterative process. Monte Carlo means we have random starting positions, many of them. Energy functions mean we have some way to evaluate the plausibility of that structure. That's the Gibbs free energy function. And we iterate because we're going to go through multiple generations of these models to see what the winners from the winners from the winners look like. And hopefully by that time, you've gotten to a place after, say, 12 iterations, where you have a structure that has a really low Gibbs free energy compared to any that you looked at in the first generation. Okay, so this represented the Rosetta approach, and I'm just realizing that the first author of the 2020 paper was a guy who worked in my lab for a bit. It's just so embarrassing. I should have recognized his name at the first point. He was an intern with me way back when. Huh. All right. Now, something amazing happened in 2014. Has anyone heard of CASP? Okay. The Critical Analysis of Structure Predictions. It's a community assessment, let's say. So the people in the protein structure community wanted to have sort of an outside broker to tell them which of the approaches being used in a given year are more successful and which are less successful in determining structure. It's all fine and good to say we've got the greatest structural prediction software in the world, but to be able to prove it is a different thing. So among the community that's developing crystal structures and putting them into PDB, they hold back some of the information for a set of new structures once every 2 years. And for this structure that's held out, they then give the sequence to the community of structural predictors and say, what do you think this should look like? And people burn a lot of computer time at a lot of campuses and send back we're very sure that these structures are what are adopted by these particular protein sequences. And in 2014, something kind of magical happened. There had been a team from Google, Google DeepMind, that had performed in the CASP studies in prior years, but they tried out their DeepMind 2 model for doing structural predictions, and they made everybody else in the field look like they were on tricycles. It was amazing. the CASP 2014 result is clearly AlphaFold 2 and everybody else, and it's quite clear who had egg on their face. So why did they win? I would say that they won because using an artificial neural network with lots and lots of layers, yes, it requires you to feed it a lot of data to train it, to feed a deep learning model. But having these many layers within the neural network that's used allows the software to store concepts, basically. To store, this is what a beta barrel looks like, just to use an example. That kind of information was possible because this model was trained on the huge pile of existing structures that we had, and so the neural network that it was using to fold up the proteins had quite a lot of information to go upon to model what structures should fall out. The software had some nice innovations on embedding. Has anyone heard the term embedding used with AI? Okay, if you're an AI researcher in the audience, I apologize right away. I know I've already said terrible things about AI, but one of the valuable things that they did was find a good way to embed information relating to a multiple sequence alignment and provide that to a deep neural network. So when we see stuff like this is a way that multiple sequence alignments representing multiple variants of this sequence could be passed to the DeepMind software. Okay, so one of the valuable things that you can see when you look at the DeepMind site is how does a protein conformation predicted by the software change as it iterates, as it goes through a new generation based on what it learned from the past energy minimization that it performed. It's not an easy model to explain, I think. But I would also say that one of the very, very valuable things that AlphaFold did, that AlphaFold's been used for, is that they partnered with the European Bioinformatics Institute. And essentially every sequence in UniProt was subjected to the AlphaFold 2 network to create a predicted structure for that sequence. So it's very frequently the case that when you go into UniProt, you can find what DeepMind thought it should look like as a structure. Is it always right? No, it is definitely not always right. Do you see a lot of structures coming from AlphaFold 2 that look like garbage? Oh yeah, there's plenty of it to be found. But it's pretty remarkable. It's pretty remarkable, and it shocked a lot of people in 2014 with just how much better the structures that came from it were than many of the people who've been triumphing in this field for a long time. Okay. One last thing I want to talk about, and that is the concept of homology modeling. We very frequently find that the structure we're interested in is in the database. We have something to work with there. There is a PDB entry for our particular sequence. But the sequence we're interested in knowing is how does the structure change if I make some targeted mutations within the sequence? What if I replace this serine with a threonine? Serine and threonine, do those have a chemical relationship? Is there a relationship between serine and threonine?
In hydroxyl group?
Hydroxyl group, very nice, good, yes. The side chain on a serine and a threonine, both of them end in an OH. So they have similar biochemistry. They're both, for example, targets for phosphorylation. So if I replace a serine with a threonine, does the structure change much? Is it likely to change its function in vivo? If I take a very long side chain, like say tryptophan, and I replace it with a glycine, am I abolishing function for this particular protein? So you'd like to know, yes, what the structure of this thing looks like, but you don't have it's not worth your time to go through trying to recrystallize this protein and send it off to an X-ray facility. You'd like to know, based on the structure we know, if we change this amino acid, how does it alter the structure? So tools like Swiss model can try to give us that. So it's a pretty simple kind of approach here. We're going to provide it our data, that's our modified sequence, and which maybe a set of PDBs we want to choose from. It's going to search among all these different templates, among all these different putative models for these protein sequences to find which is most appropriate. It'll give us some means for deciding among different possible templates. It will build a model, maybe doing some refinement to say, how do things shift if we replace this side chain with another? And it's got to have some sort of scoring function at the end of the day to say, does that structure seem plausible or not? That's the model quality estimation. So you can imagine that this is really valuable for people who are trying to do personalized medicine. If you have a protein sequence in a tumor that has some key mutational changes, it may be that those changes are just random noise, or they could be abolishing the function of that protein. So having the ability to understand how a known structure is likely to change if you alter an amino acid is very valuable information. And so Swiss model will let you do that sort of thing. It brings us to the last slide. The takeaway messages. I realize I've been talking more or less nonstop for an hour now, but hopefully this is all sort of fitting together in your mind. Structural measurement and structure prediction are essential tools for understanding the biochemistry of a protein. We don't always have an experimental model to draw from, and that's why we have de novo models for trying to produce from first principles what kind of structure is likely to fall out. Measurement is preferable when we can get it, but it's often a multi-year task from the start to the finish to get a good structure for something. Functions may be carried out by disordered regions too. The type of structural disorder, the specialty of structural disorder has become very, very interesting across all of proteomics and protein biochemistry in general. So I think that this is a real area for us all to learn some really interesting new biology. Machine learning has been there with us as long as bioinformatics has been. People have been trying to find good ways to learn models from existing measurements to inform future predictions since the beginning of bioinformatics. And it's amazing to me what is achievable now via machine learning in bioinformatics. And I'm a skeptic about this stuff, if you haven't picked that up. We have so many different possible sequences to work from, and yet the number of folds that we see doesn't seem to be rising as quickly as the number of new structures. If you look at the number of new folds in something like SCOP versus the number of new sequences, getting a new structure doesn't always give us a new fold that we've never seen before. There are lots of times where we're generating new data for previously known folds. So there's going to be this trade-off for a while that as we develop new structures, we're going to see that the coverage of possible folds has gotten better and better over time. Do we need to generate a new crystal structure for every new protein sequence? Definitely not. Because very frequently it's possible to say, this new sequence relates to a very well understood fold. So I hope that this has been useful to you. I will try to put this video on YouTube tonight. It's just under my name, David Tab, on YouTube, so it's not hard to find. But I'm going to turn off the camera so you can ask me all the questions you want without your voice being on camera, okay?